So core practical 3 is to make a temporary squash preparation of a root tip to show the stages of mitosis in the meristem under the light microscope. So in this core practical, what you do is you take the tip of a growing root, probably from garlic, and that's because there's a meristem there. Now, a meristem is an area where loads of growth is happening, lots of mitosis will be happening there. So you remove the tips of some garlic roots and you carry out a staining procedure in order to be able to see those chromosomes. The cells are sort of frozen in time, okay? You're not going to see mitosis live happening underneath the microscope. You're going to see cells at different stages depending on when they were, what they were doing when they were frozen in time at that point, okay? In terms of the method, essentially you take those little root tips and you put them in some 1 mole of hydrochloric acid at about 60 degrees C for 5 minutes. Then you rinse them thoroughly with tap water, pat them dry with some filter paper, and you take a mounted needle to then put them onto a slide. You cut off the 2 mil of the tips that you need and you discard the rest. And then you added a couple of drops of this stain called acetic aursine, which is going to stain the chromosomes a nice sort of red colour. Now we need to make the squash. This practical is called a root tip squash because we're going to take the root tips and we're going to squash them. And that's because if you just look at the root tip on its own, it'll be too thick and chunky down the right microscope. We need to squash it to get a nice thin layer of all the cells. Now to do that, you squash them down with the thick end of the mounted needle. You put a cover slip over the top and you wrap it in filter paper and you squash it together, and that will push the cells out, like I said, to make a nice thin layer. Okay, you're then going to view it under the light microscope at 400 times magnification, and you're looking for these blocky cells which show the meristem. And you can work out something called the Mitotic Index by looking at the number of cells that are in mitosis over the total number of cells that you've counted, and that gives you an idea of how much mitosis is going on there. And you should be able to see, like in this picture here, cells that are in the different phases, metaphase and anaphase, really clearly, hopefully some telophase as well.